Okay, I'm waiting for the questions and for the joining. Okay, starting with few questions I have got to be answered. Uh, start starting with first question. I'm starting with the MOET of chapter 9. That is, we have already studied about the MOET, multiple ovulation. So it is multiple ovulation transfer technology. So multiple ovulation transfer technology, if it is visible, is this visible? Okay. So in MOET technique, what we do, we take a female cow. We take a female cow. This female cow will be genetically, it will be, we can say that it will be very qualityful. It will have genetically, so that that type of cow which will be genetically more advanced, that type of cow we will take. And this cow, female cow, in this we will administer the hormone. This hormone will be administered with FHS like activity. Now we know that FHS like activity means follicular stimulating hormone. Follicular stimulating hormone, the role of this hormone is to stimulate the follicles. Along with that follicles, it develops the ovules also, of eggs also. Now these hormones will result into the super ovulation. Now what is super ovulation? Super ovulation means normal ovulation, in this generally one egg is produced, but in super ovulation, in per cycle 6 to 8 eggs are producing. 6 to 8 eggs it produced. So in super ovulation, this type of female cow, after the administration of hormones injection, it will produce 6 to 8 eggs. These 6 to 8 eggs will, when it will produce, after that, it will be mated with any allied bull, or artificial insemination will be done. Artificial insemination will be done. Now, mating with allied bull means that will also be have super genetical combination. Now, after this, the both male and female gamete will have a good quality of the genetic number, and it will produce a good uh, offspring. Therefore, it is chosen as a live bull or artificial insemination, which will, where semen will be of like a live bull only. After this, it will be fertilized. A fertilized egg, now it will be retrieved. The fertilized egg will be retrieved. This retrieved fertilized egg will be taken at 8 to 32 cell stage. We know that 8 to 32 cell stage is the stage of blastomere. So at this stage, it will be taken, and 8 to 32 cell stage, after this, when it will be taken, this will be transferred to the surrogate mother, right? This will be transferred to the surrogate mother. After the transferring of into the surrogate mother, it will be develop. It will develop and it will grow the after the time period or gestation period. It will develop a baby. It will grow a baby, and then again this same genetic mother, the female cow, where we have administered the hormone, this will be active for the next super ovulation. So that again we will use the genetic mother. So this is what we called it as MOET, multiple ovulation transfer technology. Means multiple ovulation we are doing, that is super ovulation, and then we are transferring it into the surrogate mother. We are transferring it to the surrogate mother, right? So this is the doubt which you have asked about the MOET. I just want to know, is this clear? Is this clear? You can give reply. If it is clear, you can give reply to me in WhatsApp. And moving to the next topic, that is, if it is clear, I'm moving to the next topic. That is, you have asked me about the acquired immunity. Acquired immunity. If we see acquired immunity, the term acquired self referring that we acquire something, right? We acquire something. What we are acquiring? We acquiring the immunity. That's why we say that this type of immunity is acquired immunity is uh, obtained after birth. We obtain after birth. right? This type of immunity we obtain after birth. So that is what acquired immunity. Now, now this immunity, why it is so special? This immunity, why it is so special? Because it has some characteristics. If we see the characteristics, if we see the characteristics, we will find that it has four characteristics. It has four characteristics. One is it is specificity. It has specificity, means it particularly work on particular microorganisms or pathogens. So it is very specific. It is having a property called discrimination. Discrimination. In discrimination, what they do, they identify each and every pathogens separately, and and it is also discriminate between the self and non-self. It is discriminating between the self and non-self. That means we can say that ours, which is our self and which is other self, that will be discriminated by the immunity. That is the property we can find. And also we find that it is having a property, another property or characteristics, that is diversity. So it is very diverse. We find enormous number of acquired immunity, right? Acquired immune cells. And along with that, we find another properties that is very important property, and it is memory. So they store memories also. They have memory and they store this memory for any action in the future. So these are the characteristics, these are the features we find in the acquired immunity. Now, who is controlling this acquired immunity? Who is controlling this acquired immunity? The acquired immunity is controlled by lymphocytes. These lymphocytes are mainly responsible for the acquired immunity. These lymphocytes are having two cells. One is B cell and another is T cell. B cell and T cell. They have B cell and T cell. B cell are mainly B cell are mainly responsible for the production of antibody. B cell are mainly responsible for the production of antibody. So it produced antibody. This antibody, along with the antibody, it also produced memory cell. It also produced memory cell, right? It also produced memory cell. So B cell, it produced antibody and memory cell, right? Memory cell will be used in future, and the antibody will be against the, it will be used against the antigen, right? It will be used against the antigen, right? Clear? Now, there is T cell. T cell, the role of T cell, function of T cell is that it will directly it will be involved in the uh, killing of pathogens. Killing of pathogens. Why? Why? The reason is this T cell, these T cells are having surface receptors. They have surface receptors and antibodies, antigen binding site. It is having surface receptors and antigen binding sites, which helps in killing directly the binding sites, which helps in killing directly the antigens, right? So that's why this acquired immunity, we can say, one is B cell, which is doing humoral immune response, humoral immunity. It is providing humoral immunity. Humoral immunity means, it is secretion of the immune cells into the body fluid or the plasma, and this is cell mediated immunity. This is T cell is doing cell mediated immunity. Cell mediated immunity. So these are the acquired immunity, right? Acquired immunity. Now, if anyone is having any doubt, you can ask.